and molecular biology from the University of Cagliari in Italy. She did complete her PhD in Germany, and from September 2023 she joined the lab of Professor Thomas Jacobs in plant editing, and she is now working on the Omega project. So Silvia, I give you the floor to tell more about this project.
Okay, thank you. So I can start. So thank you, Sarah, for the nice introduction, and hello, everyone. So today I'd like to introduce, is it working? Yeah. I would like to introduce you to the project I've been working on as part of the group of Thomas Jacobs, the Plant Genome Editing Group. And we are located in Ghent, in Belgium. And the focus of the group is the development and optimization of genome editing tools and synthetic biology in plants. So I would start with an broad introduction about synthetic genomics, which is a field within synthetic biology that aims to engineer novel life forms by synthesizing entire chromosomes and genomes. This is a field that has advanced from simple genetic modification to the groundbreaking creation of the synthetic yeast genome. And this progress can increase our understanding of genome architecture. Therefore, extending synthetic biology to plants is vital. Although it's challenging, of course, it's difficult because plants clearly have more complex genomes than yeast. However, synthetic genomics offers a powerful tool to address key questions in plant biology, unlocking application like cost-effective metabolites production and the development of more efficient and resistant plant varieties, all innovations that are essential for adapting to environmental changes and ensure robust food production. So the project I've been working on is called Omega, which stands for Orthogenic Mega-Base Engineering of Genome Architecture. The aim is to develop technologies to engineer plant genomes on a larger scale. But to do this, we need experimentally determined lists of essential genes, which are not currently available in plants, but have been crucial for such large scale projects. In startup mutagenesis screens, functional redundancy is the limit. It's our limit to the ability to determine gene functions. However, with CRISPR screens, now we can overcome this problem. So the Omega project consists of 4 work packages. I've been working on the first one, the genome-wide CRISPR screen to determine essential genes, together with the amazing Omega team. Julia, a PhD student, working on the work package 2 and 3, together with the technician Ian, who is also now working on the package 4. And Bram, our former technician and now a PhD student in another lab, and our new technician, Thais. So, but how does the CRISPR system work? The CRISPR system, CRISPR-Cas9, have revolutionized undoubtedly functional genomics studies. But essentially, the CRISPR system, especially CRISPR-Cas9 system, may be thought of as a programmable restriction enzyme system. It consists of two parts, a guide RNA here, and the endonuclease Cas9 that cuts both strands of the DNA. So the guide RNA has a specific 20 nucleotide targeting sequence, which is the programmable part that identifies the genomic target And this is linked to a sequence necessary for the recruitment and the stability of the Cas9. When the target is identified, the presence of this sequence, the PAM, which is in the host genome, for Cas9 is NGG, allows Cas9 to create a double-strand break 3 or 4 nucleotides upstream of this PAM sequence. Now, the mutagenic effect of Cas9 comes from the repairing mechanism of the cell. And the double-strand break is normally repaired by the cell via the non-homologous end-joining repair pathway, which is prone to error and therefore can introduce mutations like deletions, insertions. Another route is the homology-directed repair, which happens when a donor DNA with homology sequences flanking the cut size is provided, and therefore the precise correction can be achieved. Now, CRISPR knockout screens can be used to functionally characterize genes at a whole genome scale. The guide RNA provides a specificity to this tool, so this flexibility enables the full CRISPR screens to be performed. And in plant CRISPR knockout screens, the plasmid pool can be introduced via agrobacterium or biologist transformation, and therefore generating mutants collections that can be screened via sequencing. So a key advantage of the CRISPR screens is their ability to produce higher order mutants, overcoming therefore the functional redundancy. So with the CRISPR knockout screens, we can identify essential genes and genetic interactions. In the Omega project, we work with the moss Physcomitrium patens as our model organism. Working with Physco has certain advantages. For example, it grows fast, it's easy to cultivate, it has a high rate of homologous recombination, which is pretty unique in plants and comparable to yeast. And being a moss, it spends the majority of its life cycle in an haploid phase, which means that mutations are manifested immediately. We can also count on efficient and validated transfection and regeneration protocols, and CRISPR has also been already applied. So this is a bit of the layout of how we are organizing our CRISPR screen. So individual moss lines are generated with knockout mutations in clusters of around 10 genes at a time. 2 guide RNAs are designed per gene, together with the primers for the screening later on. And we use a software that was developed in the lab by a colleague, which is the SMAP design. Then the Cas9 and the guide RNAs are transfected into protoplast via PEG-mediated transfection. And from each transfection, the regenerated plants are isolated and then genotyped for the target genes. The genotyping is performed via multiple amplicon sequencing, so via NGS, on the target genes. And when a gene cluster contains plants that do not regenerate, or only in frame or wild-type sequences are recovered for a given gene, then that transfection we considered to be an essential gene hit. Hits could be due to knockout of an essential gene or ineffective guide RNAs, of course. But if in-frame mutation can be observed, then this can confirm that the guide RNA was active and supported the essentiality of the targeted gene. So, and the 10 targeted genes then from the essential gene hits, they can be systematically knocked out in different combinations to identify the causative gene or genes and confirm then the results from the primary screen. Physco has around 36,000 genes in total. So targeting around 10 genes at a time serves two main purposes. Of course, it reduces the number of independent transfections that we have to perform. So it's still a big number, but definitely lower than 3,600 instead of 36,000. And it also has the potential to reveal most of the genetic interactions between genes. So I would like to spend just a few more words about our transfection pipeline as an overview. And one of the advantages really that we have working with Physco is that from the day of the transfection, after 1.5 months or maximum 2 months, we can already have mutants grown enough for the screening. So it really doesn't take that long. For the optimization's purposes, for example, to optimize our the editing efficiency of our Cas9 architectures, we can count on the APT gene, which is an endogenous marker. And it works like this. When the APT gene is mutated, only the mutants can survive on a media that is supplemented with a 2-fluoroadenosine. So therefore, we can immediately see if the Cas9 and our guide RNA was effective in introducing the mutations, because the wild type won't survive in this 2FA medium. And also, we can check the transfection efficiency via GFP expression if we provide a reporter gene, a reporter plasmid, and also via the resistance to the antibiotic cassette that is integrated here in the plasmid, bearing also Cas9. So to conclude this overview, I would like to emphasize the potential of genome editing technologies to improve a wide range of plants. In the Plant Genome Editing Lab, we work to develop these technologies, and the CRISPR screens will definitely expand our understanding of plant gene function. So I would like now to thank my group, the Plant Genome Editing Group, in particular, Thomas Jacobs and the Omega team, and thank you all for your attention.
Thank you, Silvia, for this clear and very interesting presentation. Again, we encourage the participant to keep their question for the second part of the session, or drop them in the chat, and we will report them during the Q&A. So now we will move to Ethiopia with Hewan to discover how this tool and technology are used on tropical crop production. So Hewan is an associate dean and assistant professor at Hawassa University. She is specialized in biotechnological tools for crop improvement. She holds a PhD from Hawassa University in Japan, where she worked on the identification of genomic region associated to drought in teff. She did a postdoctoral research at the Israel Agricultural Research Organization, and in 2024, she became an Open Door Fellow. So she started her collaboration with the University of Ghent and VIB on genome editing technique to enhance sustainable tropical crop production. So again, Hewan, the floor is yours.
Okay, can you hear me, please? Thank you.
Yes.
Okay, thank you very much for giving me this opportunity. Since Silvia, she started introducing about the CRISPR-Cas9 genome editing technology, I'll go directly how this technology can enhance our breeding effort in Ethiopia and around. First of all, as you know, the world has a lot of challenges. According to the FAO information, we have to increase food production by 2030 and water by 30% according to IFRI, and also energy by 50% according to the IEA information. So this is all aggravated by climate change. And climate specifically affects it increases acidic soil throughout the world. As you can see, all this part of the world in red is affected by soil acid. So Ethiopia is not exceptional, and this region in red throughout Ethiopia is affected by acidic soil. And you have to understand that this place is the breadbasket of the country. And this is really a disaster. And when we analyze the problem of acidic soil in Ethiopia, 4 reasons are the major factors: the parental material of the soil in our region, and the heavy rainfall, and the use of acid-forming fertilizers and the removal of crop residues from the agriculture feed are the major reasons. And when we look at the problem of this, the harmful effect of acid soil is either it increases or decreases certain elements in the soil. For example, it significantly reduces nitrogen, sulfur, and potassium, which is essential for plant survival and growth and development. And also it increases like iron, manganese, copper, and aluminum, which are needed only in trace amount, but affects the root development and also the overall structure of the plant growth. So in the presence of these heavy metals, plant will not become productive, and their productivity significantly reduces. So when we look at the response of living organism for acidic soil, when we look at human beings, we move from one place to the other. And I brought here an example. Insect is one of our home garden crop, which we rely, 20 million people rely on it. And we have no understanding whether it is tolerant or susceptible to this acidic soil. But definitely we know kale, this is also one of our agroforestry crop, is tolerant for acidic problem, but not barley. So this tells us some cereals are susceptible to this acidic soil, and we compare the acidic tolerance of different cereal crops. So for example, finger millet and certain rice varieties, they are tolerant for acidic stress in the soil, and maize and sorghum are moderately tolerant. So you might ask me then, why not you grow these highly tolerant and moderately tolerant crops? But these crops mainly grow in the low altitude in our country. And when you look at wheat and oats and barley, they are highly sensitive to this acidic soil. And specifically, barley grows only in the highland area where the temperature is very cold. And that is the only cereal for these smallholder farmers. And we try to understand if why barley, because barley is an ancient domesticated plant, 9,000 years ago. And specifically in Ethiopia, it is a deer crop, and it is a cultural crop variety where different foods and drinks can be made. And also it is a feed for the animal, and it's also a construction material for the tukul, for the farmers who are living in our country. So barley has adaptive mechanism because of the diversity. So some of the adaptive mechanism is either by metal detoxification, oxidative stress management, and enhancing nutrient adaptation because of the root adaptation. So we understand that there is a certain type of tolerance mechanism. For example, I wanted to clarify for you one point about metal detoxification in the case of barley. So here, the genomic structure of barley, 2 types of barley, Morex and Murasaki mochi. Morex is known for acid susceptibility, while Murasaki mochi is understood as acid-tolerant barley. So you can see here, HVST1 gene, it is known that for releasing citrates in the root tip, and it is more expressed under aluminum stress. So this gene, as you can see, the structure is the same for both for Morex and Murasaki mochi. The difference between the tolerant and the susceptible one is because of the presence of this multiple transcription start site insertion as a kind of transposable element. So the genomic structure of Murasaki mochi, because it has 1 kilobase per insertion, it is tolerant for acidic soil, it serves as a promoter. So as you can see from this gel image, the aluminum tolerant cultivar and aluminum sensitive cultivar, they are different because of the presence of insertion. Then we have tried to see how this 1 kilobase per insertion is abundant throughout the world. So this paper indicates that 1 kilobase per insertion in barley for HVST gene, which is tolerant for aluminum-induced stress, is found in the Southeast Asia, in the Asian country, Japan, China, and Korea, where acidic soil, the genesis of the soil is starting from the acidic. But we didn't find it in Europe, and also none of the barley have 1 kilobase per insertion in Ethiopia. So the next step that we did in our country is to understand what type of barley they have in Ethiopia, because the smallholder farmer, we know that they have more than one type of land races in their own garden. And as you have to be reminded that in Ethiopia, the majority of the farmers, they have less than 1 hectare land for all their cultivation. So they maintain one or more than 2 varieties or land races of barley. So we understand that they maintain it because of its color, drought resistance, and taste of the barley in different purposes. So we did research, and we selected barley lines, and we presented to the farmers, the smallholder farmers, and the majority of them, they are specific about what type of barley they really wanted. They wanted a higher grain yield, a straw yield, white color, and large seed. So that indicates that if we give them higher grain yield, but if the color is not fit, they might not select it. So this is a very good indication. So for acid stress, which is really devastating in our region, so we have to breed. And for breeding, we have to select the varieties if there is some kind of diversity. We use the molecular markers, the genome sequencing, and we identify that actually Ethiopia has a number of diversity. So a significant population structure with different ancestral pattern. So this diversity can be a starting point for us to search for that. And when we checked the presence of insertion, none of this barley have the 1 kilobase per insertion. So that means we can give up whether they cannot be acid tolerant. But we didn't do that. Rather, we did 2 strategies. One, breeding, and the other one is searching for acid tolerant barley. In our breeding effort with Ayalnesh, who is now working at another university, we use Murasaki mochi. This is a Japanese landrace, which is already known as a tolerant barley, because we have to breed it because it is a temperate barley. It can't grow well in Ethiopia. So the other one is the Ethiopian barley that we selected. As you can see, the Ethiopian barley doesn't have 1 kilobase per insertion. The Murasaki mochi has 1 kilobase per insertion. So we are successful breeding by using marker-assisted selection. But the problem happened because the farmers, they were not happy, because they are not happy about the trait that we are using them. It become hairless, it become violet color, and they are not happy about that. So we have to search for the acid-tolerant mechanism. And luckily, when we are using the gene for transcription and transcription analysis for the relative expression of this HVAST1, which releases citrates in the root tip, we identify that comparable to Murasaki mochi, the 2 are tolerant. But What is the tolerant mechanism? We have no idea. So we might end up with a problem if we just use as it is. So this indicates that we have to develop a tool. Because traditional breeding, it involves, it takes more than 40,000 genes. It takes 8 up to 12 years. It is less precise and it has high land cost. Even I terminated my research because of after 4 years, I can't afford to pay for the land price, and unpredictable traits are there. But if we use genome editing with CRISPR, it's just a few DNA letters are affected. It takes 3 to 4 years and it is precise, low cost and predictable. So this indicates that that's why I have been involved with the Open Door Fellowship, where I have been assigned in Thomas Jacobs laboratory, working with Silvia, learning on the with the Omega group about the genome editing technology. So this takes me only 2.5 months in order to learn all the techniques and from start to the finish line. So what is the take-home message here? Diversity is a potential. So plant diversity tells us that some varieties might be naturally acidic tolerant. However, the underlying mechanism of this tolerance remain unknown. And current breeding technology is inefficient, specifically in addressing acidic soil in our country. So there is a need for advancement in genome editing by using whole genome sequencing and CRISPR-Cas9. So this approach will facilitate and for easier plant selection and improving breeding strategies, specifically in the current problem of climate change, which we have to aggressively increase food productivity and use all the land resources we have. So first of all, I really like to thank the Open Door Fellowship for helping me to establish a connection with the Genome Editing Laboratory, with Thomas Jacobs Laboratory, which helped me to learn the technology and plan to transfer this technology to Hawassa University. And thank you, Thomas Jacob, for helping me and hosting me in the laboratory with vibrant laboratory, amazing, talented laboratory members who helped me to learn every technique within a short period of time. And they really gave me a lasting impression about how laboratory science can be done in a fun way and in a transparent way. And thank you very much for different laboratory members who are working with me, Ayan Nesh Takwam, who did their master, and now they are really influential in different laboratories. And also currently I'm working with Salam, who's working for this understanding the mechanism for drought, acid tolerance in barley, and also the farmers who really gave us the land. And thank you very much for a lovely friendship with Silvia and Anjali and Nancy when I was staying there, and also the different vibrant, lively group at Thomas Jacobs Laboratory and everything. And thank you very much.
Thank you, Hewan. Yeah, it's great to see how the technology developed on a plant model are used to fight world challenges. So we can now move to the second part of the meeting. The participants are invited to ask a question they might have. We already had a few in the chat, so we can start with those. Elias from Kenya is asking, how do you design your gRNA to ensure specificity and reduce off targets? And how do you go about designing gRNAs for new species whose genetic information isn't available in a database? I think this targets more Silvia's presentation, but Ewan, feel free to also add.
So thank you for the questions. So how to design the guide RNA for specificity and reducing off targets? We use this SMAP design, but the SMAP design is based on web tools that are available, like CRISPR, for example. And in there, you can customize certain parameters. So you can choose then amongst certain amount of available guide RNAs, potential guide RNAs, because of the PAM sequence presence in the genome. You can choose those that fit mostly in the specificity score and the less off targets. And then designing guide RNAs, if you don't have the genetic information, I would say then the first step is not the design of the guide RNA, but to retrieve, to generate the sequence, the genomic sequence, de novo in this case. And afterwards, one can apply, the design of the guide RNAs.
Okay, perfect. Thank you very much. Another question we have from Nigusu is, can we say that genome editing is relatively cheaper compared to conventional breeding, where modern laboratory facilities are really lacking, especially in developing countries?
I would say yes. One of the groundbreaking aspects of CRISPR, apart from the elegant mechanism that it has and the simplicity, it's also that it's relatively cheaper, of course. It's cheaper in terms of consumables in a way, like the work, and also the time. In principle, like Hewan described very nicely, it really cuts the time because it's much more specific, and of course the technology itself.
Okay, perfect. Thank you very much. Now
Can I add on this one, please?
Yes, of course.
Yeah,
Sorry.
as like as might be it sounds as the modern technology, genome editing might not be applicable in our situation, but there is always open way to collaborate. So we don't need to invent the wheel, but we can create collaboration where the advanced laboratory, they can develop that and we can bring it and to work in our country. And yes, the investment will be high if you start from the scratch. But if we are hosted with those genome editing laboratory, since they have already the different tools in their hand, and it will become more easier. So then what we need is like to bring the seed and work in our country. That might facilitate it, but it will take only 3 to 4 years, which is much better than waiting for 12 years to release one variety.
Very true.
Yes, of course. Okay, thank you. We have a couple of questions for Hewan as well. So Demise is asking, what is your success from the genome editing on barley?
Okay. Actually, we have to understand this genome editing in barley is not only done at Thomas Jacobs laboratory, but there are other laboratories. For example, Hawassa University at the Institute of Plant Stress Resource, they are successfully use genome editing and to develop wheat by using the barley genome, because wheat and barley, they are from the same genome structure, the triticale group. So there is like they use information from barley to edit the gene for wheat, and it takes only about 12 months. And that is a success. And when you look at the wheat genome editing, it has A genome, B genome, D genome. It is complicated compared to that of barley, which has only one genome, 7 chromosomes. And I think the protocol is already there. I think it can be possible. So we plan to work on it, and we haven't started that, but we can't comment on that. Thank you.
Thanks. And one more question. It's from Lilay regarding the use of genome editing in Ethiopia. So what is the situation? Do you know if several projects are currently employing this technology in research in Ethiopia? And if yes, how does the current GMO policy treat genome-edited crops?
I think I have to answer that is more related to Ethiopia. Yeah, Ethiopia is now set with biotechnology. Even we are allowed to do the genetic engineering. There is the ethical cases have been, the biosafety rules and everything has been done through the organization. We have a very successful organization nowadays, and it is really working on that towards that. And we have a roadmap to do the genome editing in Ethiopia. And even now we have been through the ministry, like we are including the genome editing in our curriculum. That indicates that is a green light for us to proceed. And genome editing already started. For example, at Hawassa University, they are working with Brassica carinata, and they are working with sorghum. And some people are from Ethiopian Agriculture Research Institute, they are working on TEF. And so even there are a number of publications which is really coming out. Definitely Ethiopia is really embracing genome editing for the future. That is the way to go for the future.
Okay, perfect. Thank you. Yes, indeed, the innovation needs the support from the policy and the government to really make an impact on life of people. Okay, so when we still have a few minutes, so if there are more questions, you can write them in the chat. I think there is one in the chat. Yalem Tespa is asking or saying the problem of salt affected soil is very serious in my area, particularly for both food and malt barley. Farmers grow less economical crops and the eucalyptus tree replacing barley. How is that possible to work with projects like Jacobs for collaboration? Okay, well, maybe I can start answering this question. Is also a good way to introduce our program, but then of course Silvia, Hewan, or Thomas, even if you want to add, you are very welcome to. So the collaboration between Hewan and Silvia has been happening through the Open Doors Fellowship Program. This is a flagship program that we have here at IPBO. The idea is indeed to link scientists, specifically women in academia or research center from Africa to laboratory here in Belgium to give them access to facilities and tools they might not get access to in their home country. So this is one possibility. I don't know if Silvia, Thomas, or Juan wants to add something. It's a bit of a tricky question, but it's for sure very needed to keep linking the technology and the research that is done here in Belgium or even in Europe in laboratories with the research that is done in Africa in this case, because we might really have the solution or the tools to find a solution to world problem indeed.
I think I can add on that. It's like,
yes.
Yeah, the salt stress might be more abundant, might be in the lowland of Ethiopia around Afar region, and because I didn't know the person who questions that where he's working at, because for us barley is more abundant in the highland area throughout Ethiopia, except like few areas. We don't have like barley growing in very hot environment. It needs a very cold environment. So from our analysis, we understand that the highland abundant water or rain is really aggravating for acidic problem. And we didn't heard about like this barley in salt stress. So I think we can collaborate, we can develop that, and we can collaborate. The collaboration with Agriculture Research Institute, because Agriculture Research Institutes, they have a platform working by crop by crop. So then, depending on the region need and specifically on what crops they are really working, I think there is a local fund to start with. And after that, I think if the genome editing in barley is established, it will become easier to tackle also salt stress in a way. But developing the protocol is really important, and that is what we have to start. Thank you.
Thanks, Hewan, for this informative answer. We have another question in the chat. So I'm not sure I understand it. It's from Demis. What makes different? What makes it different? Genetic engineering. Maybe can you elaborate a bit on this question, because I think it's not very clear for me. I don't know, Silvia, Hewan.
No, maybe we need a little bit more detail.
Yes, so let's see. Yeah, we have a comment from Yalem Tesfa, who says that they are also working in Debre Marcos University on barley. In the meantime, if anyone wants to ask a question, feel free also to break the ice and turn on your camera and just ask.
Okay, I think he's saying that genome editing versus genetic engineering. I think Silvia, she can answer that.
Yes, indeed.
Yeah, well, they both belong to the similar field. I mean, to the same idea. Genome editing, I think we are talking about more the mutation that we are gonna impart, and engineering, it makes it's a bit more broad. Like part of the projects when we want to establish large deletions or re-engineer chromosomes. So in our project, in the Omega project, both are achieved with CRISPR in any case. And they, of course, they have different, they answer different questions.
Okay, perfect, thanks. Okay, another question from the chat. That's for both of you. How do you think scientists can help to share with the public the importance of this technology to advance breeding and fight climate change and other challenges?
Yeah, with a nice communication, more collaboration, more projects like this one, for example, and a lot. I would say, actually, I'm repeating myself, but a lot of communication, also meant to explain really in an accessible way to people the advantages, and in a very open and transparent way as well, so that people are not scared, or some people might have prejudice, or maybe they think it's really difficult to understand or unapproachable. But I would say with a proper communication, it can be overcome. And collaborations around the world like this one, definitely. And we need to share information, to share different stories. yes, to broaden the impact.
Okay, to add on it, yes, I think scientists, they can play a crucial role in bringing the gap between advanced technologies and public understanding, because we have to communicate effectively. That is really important. The importance of breeding technologies. We all know that there is breeding. Many people, they don't get frightened when the word breeding is talked, but with different technologies, some people, they really get afraid. But for that, a proper communication strategy has to be done. Like, I suggest engaging in storytelling. Scientists can share compelling stories about how this technology improves food security and resilience to climate change. A real-life example of success stories can help the public relate to and appreciate the impact. And public outreach and education. I think education is really important. Hosting workshops, webinars, and public lectures can provide a platform for scientists to explain the science behind breeding, the technologies, and simplifying complex concepts into understanding terms, because most of us, we might hide in our jargon-oriented language of science, but that has to be properly addressed to the people with the language they understand. And collaborating with the media and working with journalists and media outlets can help disseminate this information. So if it is repeated, many people, they will understand it to a broader audience. And scientists can provide accurate information in that case. And social media campaign and partnership with local communities, that's really important, because collaborating with farmers, communities, leaders, and local organizations is also help. And yeah, we have to add in education curriculum, integrating information about breeding technologies is really important. And also the last is about policy advocacy. Scientists can work with policymakers to advocate for supportive policies and the funding for research and development in this area. By taking these steps, I think scientists can foster a better understanding and appreciation of these breeding technologies among public, ultimately driving support for the development and implementation. That is my adding.
Okay, thank you very much. Okay, we have still quite some question. So Esperance might have a question, but I don't see her anymore. She can write it in the chat. Yes. Okay. Then I will look for a question. Yes. Okay. So Yalem Tesfa is asking, I guess, to Hewan, how do you plan to scale up your project?
Yes, we are working on that. Definitely for this project, as you know, we have to get fund, and accessing fund is really important. And definitely Hawassa University, we have a basic plant biotechnology laboratory, not the advanced genome editing laboratory, because still now the policy of Ethiopian government, it needs laboratory biosafety law 1 or 2 laboratory up to that level. And in Ethiopia, we have only 1 laboratory which really fulfill to that level of biosafety. So for us, we can't jump on working in our university. We have to collaborate. And for me, collaboration and using the already hand-on experience from other laboratory, like Thomas Jacobs laboratory, because they are working a lot on developing tools. Okay, so then such kind of thing, it might help to bring that technology. And in the process, we can develop our capacity. It is not a 1 day or a 1 night or 1 year experience. It has to go slowly and learning, and also putting into everything into perspective.
Okay, thanks. I think that's now so kind of answer to the question of Esperance. She was asking if your university in Ethiopia has all the facility for the technique on barley, especially when you compared it with your experience at Thomas Jacobs lab here in Belgium.
Definitely no, because one thing which makes it Thomas Jacobs laboratory to put a lasting impression for me is the way the laboratory arranged. It's like, it is one of the successful laboratory where everything is there to experience, do the job rather than get stressed with other things. For example, here in Ethiopia, if I wanted to work, I was afraid today also, like, the light might go on and what am I going to do for this presentation? So the fluctuation of power is really one thing that you didn't find it in Thomas Jacobs laboratory. And also, like, the supply, the way the laboratory, they arranged the supply management, they can buy like consumables in a stock, and then it can reduce the price. Management, and it's really important. And that, I think, we are not there yet in Ethiopia. We have a lot to learn from them to manage our resources, but we have to collaborate with them to learn everything. Yeah.
Okay, thank you. Still from the chat, we have Jetalev that asking Hewan, as you have said earlier, we have abundant drought-tolerant diverse landrace of barley to exploit. So what could be your main reason or perspective to focus on genome editing instead of other means of improvement like hybridization or crossing, etc.?
Yeah, it's like the diversity is really a lot, yes, in Ethiopia, because we have to understand that Ethiopia is, first it has been considered as a center of origin for barley, but later on they changed it into a center of diversity. And from my research, I have identified a lot of diversity. We have ample resources. But like, from my experience, Ayalna, who was my master student working with me, it took her 4 years to finish her master studies, because the first thing is like, the resources are not available there. And then we have to rent a land because in Hawassa University, we are in above about 1,700 meters above sea level. But, majority of barley growers are like, above 2,000 meters sea level, which is a little bit far from us. And we have to move through that places. The transportation cost is there, and a lot of costs are there. And we have to also rent the land. And sometimes farmers, they might not be happy to be, they can use their small plot of land to do a lot of things. And can imagine the majority of the farmers in Ethiopia, they have about less than 1 hectare land for everything. So now if we ask them like, okay, can you rent your land? That's really like, okay, can you stay empty stomach for 1 year and like use your land for research? I think that is not ethical. Yes, we tried it, and even when we breed acid-tolerant barley, we gave the acid-tolerant breeding result for Poleta research, where they can release it in the near future, because after selection, definitely, because unwanted traits happen. You see, so such kind of breeding is really good, but it involves more than 40,000 genes, because like the barley genome is about 5 gigabase pair, and the number of genes it has about, like in Moses is about 35, but in barley it's about 40,000 genes. Can you imagine working with 40,000 genes, and by breeding it is like mingling everything. So that is why genome editing is really precise. So since you are not working on the actual plant, but through the genome, you can select which genes that you can target. And it is pinpointing towards that direction. So I think genome editing is more advanced than traditional breeding, where we can reduce the cost of land and the cost of time. For example, if you employ one person for breeding for 12 years, you have to pay them. That is a lot. And you need water, you need land, you need fertilizer, so many things. But in genome editing, you can do that within 4 years. It's like it takes about 8 years of paying salary. Even that is really advancement. And the urgency is there. The urgency is what? The land is not increasing. And as you know from our culture, from our current endeavor, many of the agriculture lands are taken by like building different things. Okay, so the agricultural lands are really shrinking. So, but the population is not reducing. And all this big population, we have been about 90 million. Now we are 130 million. I think we are like the largest in sub-Saharan Africa. So we have to understand that there is urgency to increase food productivity. So if we don't apply or employ this technology, I don't know what kind of disaster we might encounter in the future. So as a government, as a responsible people, we have to think out of the box and start collaborating and bring this technology to rescue our society.
Thank you, Hewan. Very clear. I suggest we take this last one question from the chat. So Zerex is asking, is there any field trial? And do you need to edit for color as well? Or how do you plan to go with the edited variety? And how about genome editing prospect in plant protection? So let's say 3 questions. So yeah.
Silvia, go ahead, please. You can answer specifically about like edit color and other things. Because through the EMOGA, like different gene selection might help. You can explain that, I think.
Yeah, well, you can, if you know which gene is involved for the color that you're, for the specific characteristics that you're looking for, then yes, you can do it. That's the power of the CRISPR screen or the CRISPR technology in general.
Yeah, I think regarding the field trial, I think Hewan has answered just before, right? Yeah. The power of these technologies that you don't necessarily need a field. And how about genome editing prospect in plant protection? I don't know if that's maybe beyond your area of expertise.
I think like if we understand the gene at genome level, because for example, in barley, we have a reference genome. So based on the reference genome, we can mine the genes, the different genes. For example, in Silvia's research on the moss, there is a reference genome. From that reference genome, how many genes are there? And she's curating from that gene, and she's clustering those genes based on like the different tools that she has. And we can adopt that technology. We can select the genes because the function of the genes are there. But different researches, clear literature review is really important. For example, in terms of applying this in protection. So plant protection means like, for example, fungi stress or like bacteria stress or like these biotic stresses. It might happen, and there are a number of work has been done in different croplands and doing a good literature review, understanding the mechanism, and then after that mining the genes which are responsible for that specific trait is really important. So yeah, it is possible.
Okay, thanks. Okay, let's take this very last question before we close. Gurmai is asking, with regards to barley seed color, isn't it better to have the colored ones in terms of nutrition? And why do farmers prefer white seeds? Maybe Hewan, you can answer.
I think it's like white seed is really important. That's a really interesting question. So if you give them black seed, You have to understand that acid tolerant and betel is black. Every farmer, they bring their seed to the market. And in the market, it is the market which determines the price. So if the farmer is telling, I am really suffering a lot because I am growing this in acid or drought, nobody hears that. The market determines the price and he might get a lower price. And that is not a positive feedback for the farmers. So that is why they wanted white seed for the market, because they can sell it with better price. But from my experience, the farmer who are living in the countryside, they use black seed for their own food. But they sell it the white seed because it is it is with the psychology or the market characteristics in that region. So that is one thing that we understood. I hope I answered your question.
Yes, I think it's a very interesting question indeed, and I mean, we could talk about it for very long, but let's close the online cafe for now. So thank you very much, Hewan, and thank you very much, Silvia, for your time, for sharing your research and your knowledge with us today. Thank you very much to everyone who joined. We were very happy to see that there were people from different parts of the world, and of course, this is always an enriching feature for this online cafe. We have recorded this session and we'll post it on our YouTube channel. Feel free to check it out. We will organize more online cafes, so we encourage you to follow our page IPBO on LinkedIn. We mostly advertise it there. So thank you very much once again, and see you next time. Bye bye.
Thank you.
Thank
Bye.
you. Thank you for the opportunity.
Okay.